Okay. So just do you have any doubts with any part of aerobic respiration? No. If you want me to repeat any part, or else we can move forward. Okay, cool. So that's good. So let's move forward from here. So we have completed the aerobic respiration, which is basically the also including the electron transport chain. And in today's class, what we will be starting with is something called as the, now let's see, after completing the aerobic process, what happened, what is the balance sheet. So let's check the accounts. So this topic that we are going to start today with is called respiratory balance sheet. Okay. And by respiratory balance sheet, what I mean is how much net profit or net profit of ATPs has happened due to the aerobic respiration. Now I told you that after a full oxidation of glucose, so then there is a net gain, which what is written in book says that there is a net gain of 38 ATPs, out of which 2 ATPs comes from glycolysis, the other 2 come from which? TCA, remember, for glucose. And the 34, around 34 ATPs comes from the electron transport chain, where finally the ATP synthase pump, it produces ATPs by joining ADP and PI, and which is run by the proton gradient pump, proton gradient force, right? So there's this net gain of 38 ATPs. Now the point is that, and remember that this is during aerobic respiration. Aerobic respiration, and it is per molecule of glucose. Is it clear? Is that clear, Iram? Yeah. Now this is it's a theoretically calculated number. Now what do I mean by this is, inside the body, it's not like we oxidize one glucose at a time, right? There are millions and millions of glucose molecules that we utilize in every cell every minute. So how do we come to know that there are 38 ATPs per glucose, right? So there are some assumptions that are made to find this theoretical number of 38 ATPs per glucose molecule. And this is based on just the chemical reactions, but we assume that the chemical reactions that we have studied till now inside the cell, they will all happen in that same manner, and also they will all happen one after the other in a sequential order. Because remember, this is there are many ways in which the pathway can diverge. It is not just the only thing happening in the body, which is respiratory reactions. There are metabolic reactions happening inside a plant. I also told you there is a, apart from TCA cycle, there is C3 C4 cycle, also Calvin cycle also happening. There also oxaloacetic acid and many of these substrates are used, right? So lots of things can go out and come in at different stages of this whole reaction that we studied. But write down how do we come to this 38 ATPs per glucose molecule. Write down the calculations of net gain of ATP the calculations of net gain of ATPs for every glucose molecule oxidized, for every glucose molecule oxidized is based on the following assumptions. Is based on the following assumptions. Okay. Now, the assumptions are, first, that there is a sequential order pathway of reactions. There is a sequential ordered reaction pathway of glycolysis followed by TCA cycle and ETS. Okay, so first glycolysis followed by TCA cycle and then ETS. This is the first assumption that we make, that it's happening inside the cell always in this manner. Is that clear? Yeah, good. The second assumption that we make is that, remember, we were making there was some high energy molecule intermediates produced, which were NADH and FADH. Yeah. And remember, there was NADH also produced while during glycolysis. You remember that? There was one step where NADH was produced. So the second assumption is that to make these 38 ATPs, the NADH synthesized in glycolysis also is transferred into the mitochondria. Okay, and here that NADH undergoes oxidative phosphorylation. Okay, because we are also counting that NADH which was produced during the first step here, glycolysis, that it comes and helps in producing ATPs here during the ETA. Is it clear? So write down, second assumption is the NADH synthesized, during glycolysis is transferred into the mitochondria. Is transferred into the mitochondria and undergoes oxidative phosphorylation. To contribute to ATP synthesis. So there is some share of this process also, which we have not shown directly by the chemical reactions, what we are assuming is happening, and that also contributes to ATPs forming from the mitochondria. Okay. The third assumption which we are making is that at any step which actually might happen in some disease conditions, but remember, whenever we study biology, we first study the most standard thing. So the standard is always based on some assumptions. So we say that the standard heartbeat or the blood pressure or the respiration rate of a human is XYZ, right? But we assume that the human is fit, the human is getting proper nutrition, the human is drinking enough water, is properly hydrated throughout the day, is not obese or not suffering from any ailment. That's where we make some standards, correct? So here also, when we say 38 ATPs are produced per glucose, that might vary in different disease conditions or in people who are always feeling fatigued or who gets dehydrated very fast. Okay, so in those people, there might not be production of 38 ATPs per glucose, right? But we are assuming it. Is it clear, Iram? Yeah, so the maximum it can go is till 38, taking all assumptions into consideration. So the third assumption is that none of these intermediates that are formed in any of these pathway are actually utilized to synthesize something else. So we are expecting that when one molecule of glucose enters, it gives two molecules of pyruvate. Both the molecules of pyruvate enter the aerobic pathway. They don't go and get wasted in fermentation or something else. So they enter the aerobic pathway, and then whatever intermediates they continue to form, all of them, millions of those molecules always enter the next step, which is the ideal situation. Okay, so the scenario is that none of the intermediates in the pathway are utilized to synthesize something else or synthesize any other compound. Is it clear? So Devangi has joined. Hello Devangi, how are you?
Good evening, sir. I'm fine.
Good evening. You got a little late. Everything okay? How's your health? You're keeping well?
Yes, sir. I was preparing for a test.
Okay. Okay, cool. So we just me and Iram were discussing. Hello Waziha, how are you? Am I audible, Waziha? I don't know if I'm audible to Waziha. Okay, please reply Waziha if you can hear me. Yes, okay, fine, cool. She's replying in the chat. Okay, so we were discussing in the respiration, the respiratory balance sheet, okay, Devangi and Waziha, that we get 38 ATPs per molecule of glucose during aerobic respiration, okay. And the thing is, we were discussing that how we reached to this number, because it's not like in our body every glucose molecule is oxidized one after the other. There are millions and millions of glucose molecules oxidized in every cell, in every second, every minute. So how do we come to this number? So this number is theoretically calculated based on the reactions that we know happens, okay. And there are many assumptions to this calculation, and these were the three assumptions we have discussed till now. First assumption is that whatever we have studied, our glucose molecule in actually follows that sequence or that orderly reaction pathway. First it undergoes glycolysis, then TCA cycle, and then ETS pathway. And we also, second assumption is that the NADH which was synthesized during glycolysis, okay, also reaches finally the mitochondria and undergoes oxidative phosphorylation to contribute to ATP synthesis. Is it clear, Devangi and Vazia? It's okay, Vazia. I think Iram's mic is also not working today, so you people can write in the chat. Okay. The third assumption was that none of these intermediates that we have studied in any of the pathway, glycolysis, TCA or ETS, none of these intermediates actually are lost or are utilized to synthesize any other compound. Now, these are ideal assumptions. There may be disease conditions or physiological conditions during which all of them are not, all our assumptions are not holding true. In that case, the number of ATPs per glucose molecule may decline in the body, but remember, because we have millions and billions of molecules being oxidized in every cell per minute, it's not a problem. Okay, unless and until we are doing things good. And the fourth assumption and the last assumption that we make to calculate this theoretically is that only glucose is being respired. So we are assuming that only glucose is the molecule which is broken down oxidative, which is oxidatively phosphorylated or which undergoes oxidative breakdown in the cell. No other substrate are entering the pathway at any of the intermediate stage. For example, when glucose enters, it becomes glucose 6 phosphate, and then it becomes glucose 1,6 bisphosphate. Then it converts to fructose 1,6 bisphosphate. Remember glycolysis? Thumbs up if you remember that. Yeah. So the idea is that we are assuming that the cycle always starts from glucose. By any chance or by any means of nutrition, we are not directly taking in fructose 1,6 bisphosphate, and it does not enters directly into the pathway at the fourth step. Because if in our body we already have fructose 1,6 bisphosphate, it can just enter the glycolytic pathway by just jumping the queue, right? No need to convert glucose into glucose 6 phosphate and then glucose 1,6 bisphosphate and then into fructose 1,6 bisphosphate. And that may happen under normal situations because we are eating diverse food, and that may happen, but we are assuming that only glucose is being respired. So write down, no other alternative substrate is entering the pathway at any stage, is entering the pathway at any stage. Okay, is that clear? Everyone, is that clear? Okay, so Devangi, since you can speak, do you have NCERT with you?
Yes, sir.
Yes. Vaziha and Iram, do you also have NCERT with you right now? You can write your answers. Yes, okay. Please open your NCERTs and come to page number 235. Page number 235, you have to mark a 2-marker question there. The starting of page number 235, which says comparison between fermentation and aerobic respiration. Do you find that? Everyone finds that? Devangi? Devangi, would you like to read it for us? And everyone please mark that as a 2, it can come as a 2-marker or a 3-marker question, that what is the compare or draw a parallel scheme between fermentation and aerobic respiration, or difference between fermentation or aerobic respiration. So would you read that, Devangi?
Yes, sir.
Yeah.
Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration, it is completely degraded to carbon dioxide and water.
Yes, is the first point clear? Anyone? I want 3 thumbs up if for everyone it's clear, or anyone who wants any discussion, Please write in the chat. 1, 2. Wazia, what about you? The first point that Devangi just read right now, is it clear to you? Yeah, page number 235, 235. Okay, page number 235. So Wazia could not find it. She is reaching there. Let me know when you are on page number 235, Wazia. Okay, so first sentence. So Devangi, will you please reread it?
Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration, it is completely degraded to carbon dioxide and water.
Is that point clear to you, Wazia? Or do you want any explanation or discussion on this? Clear, perfect. Second point, Devangi.
In fermentation, there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic conditions.
That also clear? So if any system undergoes fermentation, there will only be 2 molecules of net gain, and that also in the glycolytic phase, the first glycolysis, because in fermentation no ATP is produced. Wazia, Iram? I got Devangi's thumbs up. What about Wazia and Iram? Second point clear? Yes, it's clear to Iram. Clear to Wazia. Third point, Devangi.
NADH is oxidized to NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration.
Yes, is that point clear? Okay, so in fermentation, it is a slow process of NADH oxidizing to NAD plus, whereas in case of aerobic respiration, it is very, very rapid a process, because remember in TCA alone, there are around 8 NADA, 8 NAD plus molecules that are reduced, right? Make sense? And then all these NADH goes to ETH and get oxidatively phosphorylated there. Perfect. So I think the gist of respiration is clear to all of you. Now we'll do an in-class activity. I hope everyone could speak, but since only Devangi and me can speak, so we will divide it among ourselves. So just below that, there is one paragraph marked as amphibolic pathway. Can you see, all of you? Ziha, Devangi, and Ian. Amphibolic pathway. Yeah. Now, what do you mean by amphi? Amphi. Where else have you heard this word in biology? Devangi?
Amphibians.
Amphibians, right. And what does amphibians mean? Also, you must have heard of terms called amphitheater. Means again related. So amphi means dual or both. Dual. Amphibians depend on dual conditions for their life cycle, right? Water and land, correct? Amphitheater is a theater for dual cues, which is sound and vision, right? That's why it's called an amphitheater. It harnesses both sound and vision. So you can see plays in amphitheater where you are seeing someone performing, also hearing a sound, right? And then comes amphibolic. So the word bolic is from metabolism to bolic. So we know that catabolic, and there are two type of two type of metabolic pathways which we have studied till now. One is called catabolic, one is called anabolic. Catabolic means breaking down, right? Anabolic means making up. Amphibolic, on the other hand, is a pathway where both of these are happening simultaneously or in the same pathway at different stages. Do you understand that? Everyone, thumbs up. So with respiration, let me tell you the gist of this paragraph. Write down if you want to note this down. So respiration was earlier used to, scientists used to believe that respiration is a catabolic path process because it involves breaking down of glucose, a 6-carbon molecule, and this is how we define it. We are breaking it and breaking it till a point where every carbon is shredded out and teased out and converted into CO2. That's what respiration is. So we have a mind makeup that in respiration we are just breaking down and breaking down. So that's why it could be a catabolic process, right? But this understanding is not complete. Okay, we know that in respirations at different, the holistic picture of respiration is that at different stages different substrates can enter. Okay, and remember when I was defining respiration for you or aerobic respiration for you, I said that it is the process where substrates like glucose are or organic compounds are broken down or oxidized, right? Those organic compounds could also be protein, starch, etc. Need not to be glucose all the time. Do you remember that? Yeah, Devangi and Neera? So we figured out that it's better to consider respiratory pathway as an amphibolic pathway. Amphi means dual. So it also does catabolism at places. It also does anabolism, also makes things more complex at places. Okay, so we cannot just call it a pure catabolic process. It should be called an amphibolic process. Now I want all of you, all three of you, to read that paragraph taking 5 minutes, and then I will be asking you questions and explaining the flow chart which is given below. Iram says it is also called as metabolism, right? No, Iram, metabolism is where we sum up. Metabolism is defined as the sum total of all the reactions happening inside a living system, inside a cell. Okay, all catabolic, all anabolic, everything. That's metabolism, and including all, like respiration, metabolism of fat, immune system activation, any other reaction happening in the body due to inflammation, anything, your digestion, your making up of proteins to repair the DNA, everything comes under metabolism. Do you understand? There's a difference. Amphibolic is where in the same pathway you see two natures. Some part of the pathway will be catabolic and some part of the pathway will also be anabolic. So that's called an amphibolic pathway. And the sum total of all the catabolic, anabolic, amphibolic pathways happening overall in the cell is called metabolism. Is that part clear? Yeah. Any doubt? No? Okay, cool. So all three of you, let's do one thing. So I'll give you 5 minutes by the clock to read 14.6 amphibolic pathway. Okay. And then I will ask questions to you and we will discuss the figure with the flow chart which is given just below it on the next page, and everything will make sense to you. Okay. And your time starts now. Everyone is doing that? Thumbs up. Perfect. Your time starts now. Please feel free to ask if you have any doubts. This time I will not disturb you, but you can ask if you want. 5 minutes. Okay, 2 more minutes left, people. Okay, so it's done. Devangi, Waziha, and Niram, read. Devangi. Okay, so let's start with the pathway, right? So you can see, Devangi, you can speak, so we both can talk about it, maybe. So tell me, what do you understand by reading that paragraph, Devangi? Yes, Devangi.
So that the substrates other than glucose are first broken down, like fats are broken down into glycerol and fatty acids, and
Yes, so
then they
you say
degrade
that,
into.
yeah, okay, so let's also keep making this. So you say that if apart from glucose we are working with fats, then they need to be broken down into its components. So fats are made up of fatty acids and glycerol, right? This is what you're saying.
Yes, sir.
Yeah, and then
So then fatty acids degrade into acetyl coenzyme A.
yeah, so they can directly enter the whole thing at the level of acetyl coenzyme A, right? They directly can enter the Krebs cycle. Remember the Krebs cycle, the TCA cycle?
Yes, sir.
Yeah, so they can directly enter the Krebs cycle if we are using fats, not glucose. And if we are using carbohydrate, which is like, let's say anything, any simple sugar, then what happens? If we are aspiring or breaking down carbohydrates, exact thing that we studied just now will happen. All simple sugars first have to break down into glucose, and we know that glucose enters glycolysis, right? And from glycolysis comes 2 pyruvates, and then this pyruvate goes to become acetyl CoA as we studied, and then this enters the Krebs cycle, right?
Yes, sir.
And then the NADH and the FADH, NADH, FADH2 produced from the Krebs cycle then enters the electron transport chain. Clear till here? And these fatty acids, they can directly enter at this step at acetyl CoA. Is it clear, everyone? Yep. And also, so how it works is our body first prefers carbohydrates as the source of energy. So whatever we eat from that, our body always chooses carbohydrates which are simple sugars, for example glucose, lactose, and things like that, and we always degrade it first. When our carbohydrate reserves are over and finished, then we move to fats. Okay, so that's why it is said that if you want to make this transition, you must have seen obese people working very hard to reduce their weight, but still complaining that they cannot reduce their weight, right? Have you seen that? It's very difficult to reduce weight, as difficult as to gain sometimes, but gain is still easier than once you have gained and then you try to reduce it. You understand why? Because you have to make your body shift from using carbohydrate to start using fats and start breaking down fats. And now this shift needs some vigorous activity of your body. So our body only shifts to fat metabolism under conditions where we are doing strenuous exercises. Okay, where so just simply walking, you can keep walking for hours and hours a day and you might not lose weight. Okay, it's possible because your heartbeat has not elevated enough so that it can make your metabolism shift from carbohydrates to fats. Okay, so you need to do really some playing or some exercises where you're doing some cardio and your blood is rushing through your body. Simply walking at a very comfortable pace is not going to burn your fat. Okay, and even more than that, people think that there could be beverages and chemicals that you can just drink and all your fat will be burnt. So that does not work that way. You understand? Everyone understands this? If someone is starving and even the fat reserves are over, then as the last resort, body starts degrading proteins. It's the last resort. So when protein degradation starts, which means body is eating up itself, so our muscle starts breaking down and all that protein is again being broken down to amino acids to keep the brain and the vital organs alive. So that is called wastage of muscles. So if you see someone trapped and is starving in a forest or somewhere where there is no food in extreme condition, then first carbohydrate reserves will be over, they will switch to fats, fat reserves will be over, then the body will switch to proteins and you will start seeing muscle wastage. And this protein, this amino acids can directly enter the pathway at the level of pyruvic acid. Okay, pyruvates. So do you understand? At many stages, proteins, carbohydrates, and fats can come up. So the same energy that we are getting by utilizing carbohydrates, we use that energy to make proteins, our muscles, and we can use the same protein to break it down back to amino acid and pump it back into the pathway to produce more energies. Is it clear, everyone? Now do you understand why we call it an amphibolic pathway? Hiram, Devangi, and Wazia?
Sir, I had a doubt.
Yes, Devangi.
Sir,
Please.
it's written amino acids after deamination.
Yes, deamination means you are just like dehydrogenation. Okay, D means removing amines. So from proteins, you do deamination to make amino acids back, right? D means removal. Is
Okay,
it
sir.
clear? Okay. Just like dehydrogenation, deoxidation, deforestation, deamination. Okay. Now, the last part of this chapter is respiratory quotient. Now, this part is asked multiple times because it's simple, but it is still a concept. So let's talk about respiratory quotient as well. So the word respiratory quotient, you know what is a quotient? In mathematics, you must have studied it. Quotient, remainder. No? So respiratory quotient is the volume, the total volume of CO2 which is exhaled or evolved during respiration upon the total volume of O2 consumed. Is it clear? So it is called respiratory quotient or respiratory ratio. Simple. So can you find for me the respiratory quotient for the aerobic respiration as the last thing for this chapter? Now, can anyone tell me the formula for a complete respiratory reaction? Aerobic respiration. Glucose C6H12O6 plus oxygen gives CO2 plus water plus energy, right? Is it clear? Am I correct? Or I'm missing something? Now first let's balance it. Can anyone balance it for me? Devangi?
6O2.
6O2.
6CO2 and
Mhm.
6H2.
6H2O. So this is a balanced reaction, right? Now can you find the RQ for me? It is the RQ.
1.
Yes. Because here you are using 6 moles of CO2 upon 6O2, which is utilized. So 6 by 6, the volume is 1. So the respiratory quotient of a complete oxidation event is 1. Okay. If you calculate the same for a fat, then you will find for a fatty acid, which is any fatty acid, you will find that it is less than 1. So for fats and fatty acids, it is around 0.7. And for proteins, it is around 0.9, but less than 1. Now the highest RQ is for sugar, glucose. That's why that is the most preferred respiratory substrate in our living system. And let me just tell you that for living systems, respiratory questions can also sometimes be more than 1. And also we know that fats and proteins can be utilized But under normal conditions in a living system, pure proteins and pure fats are never utilized. There never comes a scenario where you utilize pure proteins or pure fats as respiratory substrates, but they can be if needed. Okay, it's very difficult because the respiratory quotient is low. Is that clear, everyone? So anyone has any doubts in this whole chapter, please go through your notes. It is a bit biochemistry heavy, reaction heavy chapter. So please go through, and I will stop here for a couple of minutes. And if you have any doubts, this is the time to ask. Please ask me your doubts. Please take a couple of minutes. If there are no doubts from this chapter, then you can write in the chat that no doubts. Iram says no doubt, everything is crystal clear. That's great. Iram, very glad to know that. We'll wait for Devangi and Waziha's response and then move forward. Take your time and then let me know. Waziha and Devangi. Waziha and Devangi, any doubts anywhere in this chapter? Please revise this chapter as much as you can. Otherwise, these pathways are not going to stay in your head. Devangi, everything is clear to you?
Yes, sir.
Yes. Waziha, clear. Okay, cool. So with this, we finish this chapter, and let me add a new page and let's start at least the basic foundation or discussion of the next chapter, which is chapter 15, plant growth and development. Okay. So how many of you have already done this in your schools? Chapter 15. Just raise your hands. This chapter has not been done. It's a part of the syllabus, right? Anyone for whom this is not in the syllabus, please let me know. I think it should be in everyone's syllabus. So this is what we'll be doing the next, and I will just discuss some foundations of it today, some important aspects of it today in the next 15-20 minutes. And then on Wednesday, a portion of it, which will be your plant growth regulators, okay, will be taken by Jayant sir. So please do not miss the class, because what he will take on, you have to ask questions to him and get it cleared, because plant growth regulators, I won't repeat again. Like, we will take doubts if you have any doubts, but not the whole part will be repeated. Is it clear, everyone? Vaziha, Devangi, and Niram? Yeah. So the plant growth regulators part will be done on Wednesday by Jahan sir. Okay. The reason being, I'll be not available at that time to take the classes. So is it okay? Vaziha, Devangi, Niram, noted the information? Perfect. So but before that, let's talk about what do we understand by plant growth and development. So certain things that we already know is plant growth is a lifetime process, right? So plants are different from animals in the way that they continue to grow and develop throughout their life till they are alive, from a seed that starts germinating till the time when a big tree falls and dies. It is growing, which is not true with animals. Animals, after one point of time, stops growth. Okay, there's no growth after one point of time. Is this thing clear, first of all? Yeah. And we also know that there are many factors associated with plant growth, which are both intrinsic and extrinsic for the development and growth of a plant. Now, intrinsic means internal. Okay. Now, for growth, plant growth, or we call it plant development, there are factors, two type of factors. One is intrinsic factors, and the other is extrinsic factors. Okay. Now, these intrinsic simply means internal factors, and extrinsic are external. And I hope you can guess some of these. Can you guess some internal and external factors for plant growth quickly? I think you can very well guess external. Anyone would like to guess? Devangi?
Nutrients, water.
Nutrients, water, just availability of sunlight, availability of CO2 to fix, and availability of oxygen to respire, correct?
Yes, sir.
All these things define the growth, and that's where the primary growth of plant happens. And intrinsic factors, what are they? Anything? Sorry, Devangi, come again.
Genes.
Yes, the plant's genetic makeup. How well is the genome of the plant? That's also where, genetically modified plants have sometimes better yield and better growth, because their genome is mutated in that way to support growth and development, right? So first is genes of the plant. Second is the cellular and the molecular pathways, any disease that is there inside of the plant, any pathogen or infection. So plants also have their plant specific pathogens and plant specific infections, right? They also have tumors and other diseases which are plant specific. So if that is there, then there will be, it will also hamper growth, or there can be stunted growth. Now this growth that we are talking about is basically measurable, right? You can measure growth because you know at what rate the plant is growing. You can just measure it. And you also know the average cell size of a plant. So in that volume, you can calculate how many cells per unit time a plant is forming. So how many cells plants is forming per hour, right? So just as an estimate, there are around 17,000 to 20,000 cells in a young maize corn, maize or the corn plant that starts growing. It grows rapidly at the rate of around 17 to 20,000 cells per hour. Okay, so it grows very fast. How many of you have seen a seed germinating? How many of you keep plants in your homes, inside your rooms, and have seen the plants grow and germinate? Yes, Devangi. So you must have seen that if you put a seed or a seed like a gram or a rajma or something in a soil, when it starts coming out, the seedling grows rapidly in the beginning, right?
Yes, sir.
Yeah, it's very fast. And the reason for that rapid growth is because plants have certain regions which are called meristematic regions. Specifically, there are root and shoot meristems, right? So meristems are of two types: lateral meristems and apical meristems. So today I'm giving you some terms that you have to go back and study. What are meristematic tissue? Write it down. What is a meristematic tissue? Types of it: apical and lateral, both in root and shoot. And second is phases of growth. As I told you, in some phases plants grow rapidly, whereas in some phases plants do not grow that rapidly. Okay, so the growth is kind of, it becomes slow. And it depends on, yes, like Iram is mentioning, temperature is also one factor that allows the plant to grow, or like that regulates the growth, makes it faster or smaller, faster or slower. Okay, and from this phase of growth we will come to the growth rates. So what is the growth rate? Growth rate. How will you define growth rate? Anyone?
Increase in growth per unit time.
Exactly. So rate means, whenever we talk about rate in science, rate simply means anything over time, right? So growth rate simply means either you take as the biomass or the cell number. So because it is possible to calculate both in plant systems easily, so you can say that it is increase in the biomass or increase in the growth or increase in the cell number per unit time in a plant. Okay, noted. Yes, now the good thing about studying growth rate is it is a very powerful way to assess other properties of living systems as well. And on top of it, growth rate can be mathematically studied, because so there are two types of growths in plant or in animals in general, like in living systems. One is called arithmetic. You know what is arithmetic? And the second is called geometric. So anyone knows the difference between these two terms, arithmetic and geometric? You must have studied it in your maths. So anything expansion in a geometric fashion or expansion in an arithmetic fashion, what does that mean? So arithmetic means if you start with 1, you give rise to 2. Yes, Iram, right. Then you give rise to 3, 4, and so on. This is arithmetic increase, right? But geometric is you start with 1, it doubles to 2, then it doubles to 4, then it doubles to 8, and so on. So every time there is a doubling. So it's like into 2 effect. Here it is plus 1 effect. Do you understand, everyone? Which one do you think is a faster, a rapid mode of growth rate? Arithmetic or geometric?
Geometric.
Geometric, right? Geometric is a faster rate because, and can you give me any example of a living system where geometric growth rate is observed? A very simple example. You all know it. It is bacterial colony or bacterial population. Okay, bacteria always divides through fission, right? Binary fission. Bacterial population growth or meristematic tissue growth, because that also divides. That's why I'm using these two terms. Okay. So these things I will teach you the next to next class, but